Thank you. Good morning. I'd like to thank the organizers for the invitation. I was asked to talk about stem cell research and cardiology, and this is a huge area, so since I only have 20 minutes, I decided to focus on one type of stem cells, human pluripotent stem cells, which we've been working in the last 10 years. And it will be a bit a different talk than the rest of the talk, somewhat futuristic. So we've been working with human pluripotent stem cells, and what I will try to tell you about is their potential for the field of regenerative medicine to develop treatments for heart failure and cardiac arrhythmias, their potential implications in disease modeling and personalized medicine, their potential use for drug screening and drug development, and if time permits, I will tell you a little bit about the new technology that we've been using called optogenetics. So the first human pluripotent stem cells that were described were human embryonic stem cells. These are cells that were derived from in vitro fertilized human embryos. The cells can be propagated in the undifferentiated state indefinitely. In fact, we've been using cells that were derived from an embryo more than 15 years ago. And when we move from these conditions, they can be coaxed to differentiate to generate any cell type in the body. And in 2001, we were very fortunate to publish the first report on establishing a cardiomyocyte differentiating system from these cells, in which spontaneously beating human heart cells were generated for the first time. Now, if one wishes to use these cells to repair the heart, it's not enough to make these cells, it's not enough that they will engraft, you want them to stay there forever and to integrate and beat in synchrony with host tissue. So to answer this question, we started with a few studies. This was an in vitro study in which we took our human heart cells, plated next to rat heart cells, and you can see that within a few hours, the two tissue types started to beat in synchrony. To verify that this is indeed true integration, we did this study on a multi-electrode array and demonstrated that there is indeed one-to-one coupling between the two tissue types. We then continued these studies with similar in vivo studies. This is one study in which we labeled our cells, implanted them in the rat heart, and 2 weeks later we studied them using an optical mapping technique. And when we looked at the maps, we can see that the cells integrated nicely with nice conduction at the area of the graft. We then performed the proof of concept studies showing whether the cells can improve the function of a heart failure model. So we created a myocardial infarct model in rats by ligating the LAD. A week later, we came back and injected our cells into the area of the scar, and we followed them over time. If you look at fractional shortening, they start here. When you create an infarct, you see immediate reduction, followed in the control group by abnormal remodeling with continuous deterioration of the function. When you transplant the human cells, our cardiomyocytes, into this area, this abnormal remodeling is prevented, and there are even some functional improvements. So this was very encouraging. So how do we continue forward? What are the obstacles in making this a clinical applications? And we recently started a collaboration with the University Health Network in Toronto with the aim of starting a first-in-man clinical trial within the next 3 to 4 years. The first obstacle is making enough cells. So our initial differentiation, only a few % became cardiomyocytes, but thanks to work of many laboratories, including of our collaborator, Gordon Keller from Toronto, we now take lessons from early development and generate directed differentiation protocol in which we could get now more than 90% of the cells becoming heart cells. As you can see in this microscope view, the entire dish is beating, and when we stain them, more than 90% of the cells become cardiomyocytes. When you couple this with bioreactors, we can now make billions of cells, and we estimate that we can make 1 billion cells, what we think needs to treat one patient, in a cost of about $2,000. So the price is getting more and more potentially applicable. The second issue is the question of immune rejection. Since we are not transplanting cells from the same patient, we are dealing with an allogeneic transplantation. Nevertheless, these cells are much less immunogenic than, let's say, an entire organ, the heart. So we think we can get away with much milder immune suppression regimen in these initial clinical trials. But the ultimate solution may be the technology introduced by this person, Shinya Yamanaka, who developed a technology called IPS in 2006, work for which he received the Nobel Prize in 2012. What Yamanaka did was to take adult somatic cells and to transiently express a set of genes that are usually expressed only in embryonic cells. By expressing these genes transiently, he was able to reprogram adult cells back to a state resembling the fertilized egg. So let's say you take somebody who's 90 years old, you take his cells and you take it back in time in a time tunnel 90 years plus 9 months back. He called these cells IPS cells, or induced pluripotent stem cells. They highly resemble embryonic stem cells and therefore can be coaxed to differentiate to any cell type. And since these cells are derived from the patient, if you transplant them, there won't be any problem of immune rejection. And this turned to be a robust technology. We routinely now take skin fibroblasts or blood cells from patients. We reprogram them to IPS cells and then use our favorite differentiation strategy to get the patient own heart cells beating in a dish. These heart cells display cardiac-specific proteins. One can perform patch clamp and show that they have cardiac-specific action potentials, and one can perform calcium imaging and show that each action potential results in calcium release from these cells. Not only that we can make it from young and healthy individual, we wanted to see whether we can make them for the patient which will actually need them. So in this study, we took a skin fibroblast from patients with advanced heart failure who were elderly and had multiple comorbidities. And despite that, we can make IPS from these patients, and we can drive their differentiation to heart cells that although their hearts were really sick, the heart cells that we generated were healthy and were young, similar to the day they were born. These can then be shown again to integrate with existing tissue and can be transplanted in animal models and shown to engraft and integrate. Now, beyond the potential in regenerative medicine, the IPS technology has enormous potential in the field of disease modeling, and especially for genetic disease modeling. The idea is if somebody has a genetic disorder, such as a cardiomyopathy or inherited arrhythmogenic syndrome. Of course, we cannot take the heart out to study it, but since the mutation exists in the DNA in every cell of the body, if we take skin cells, for example, reprogram them to make IPS cells, and then make heart cells, their heart cells should portray the same disease as the patient. To test this hypothesis, we started in a long QT syndrome in this study which was published in Nature a few years ago. We studied a young patient that came into our ICCU after she had cardiac arrest. When she came in, she had these storms of polymorphic VT. We noted that she had a prolonged QT interval, and we learned that several members of her family died at a young age. We screened the family and identified a point mutation in the KCNH2 gene responsible for the long QT type 2 syndrome. So we took some skin cells from the patients, reprogrammed them to make IPS, and then made heart cells from them. And then using patch clamp, we studied the cells. So in the top panel, you can see cardiomyocytes derived from IPS from a healthy individual. Note that we can record 3 types of action potential. One that resembles a ventricular action potential, that's the majority of cells. But some cells also express an atrial-like action potential or a sinus node-like action potential. You don't have to be a cellular electrophysiologist to see that the long QT syndrome cells express significantly prolonged action potential duration when compared to controls. So the hallmark of the disease can be manifest in the cell. Moreover, many of the cells displayed early after depolarization that led to triggered beats and even trains of triggered beats. So you had a patient coming with an arrhythmia, you took some skin cells, you ended up with her own cells in a dish having the same arrhythmia like she came. Now one can study the mechanism of this disease and also test, for example, drugs. Here, for example, we give a drug that opens an alternative potassium channel, pinacidil, which opens the KATP channels, that shortens the action potential and prevents the arrhythmia. Next, I would like to talk about a second arrhythmogenic syndrome, CPVT. There are 2 types of CPVT, CPVT type 1, which is a dominant inheritance and results from mutation in the ryanodine receptor, and we previously published our work on using IPS modeling of this disease. The less common, the recessive form, is called CPVT type 2. It's due to mutations in the major calcium buffering proteins in the SR called calsequestrin. So this is a very famous Bedouin family in the northern part of Israel. It was initially described by Michael Dar. They suffer from CPVT2 and they are being treated by a pediatric cardiologist. They are highly symptomatic and when we look at their cells, it's not surprising why. So here we're performing calcium imaging of control cells. You see nice calcium imaging, but in our CPVT population, you can see significant abnormalities as identified here. This explains why they are so symptomatic. Another interesting question that we ask is the concept of SOICR. Some people assume that there's a putative calcium sensor within the SR, that if calcium rises above this threshold, then there is spontaneous depolarization. Release of calcium will lead to arrhythmia. Normally, we are far away from this threshold. When we get adrenergic stimulation, calcium level rises in the SR, but still away from threshold. The hypothesis that in CPVT, this threshold may be altered. To test this, Leonid from my lab did a study in which he took quiescent controlled cardiomyocytes, or CPVT cardiomyocytes, and tested increasing level of calcium. You can see that normal cardiomyocytes don't have spontaneous release of calcium, only at very high doses of calcium. In contrast, the CPVT cardiomyocytes display spontaneous calcium release from the SR, even at low levels, and at high levels it's become really, really pathologic. So we think that there is some kind of interaction that involves in changes of the threshold of the ranitidine receptor in this disease, in which there is spontaneous calcium release during diastole that caused the problem. Leonid then wanted to test several drugs for their effect on the cells. Some of the drugs worsens the situation, such as adrenergic stimulants, but some help. Look, for example, an interesting drug is flecainide, which seems to improve significantly, and carvedilol. It turns out that carvedilol is the only beta blocker that also has ryanodine receptor stabilizing properties. So we wanted to see whether it correlates with the clinical status of the patient. So this is what flecainide does to the patient's own cell. You can see it eliminates the arrhythmia. What happens to the patient itself? So when you put the patient on a treadmill, you see these arrhythmias. When you treat him with flecainide, you eliminate the arrhythmias. So there's a nice clinical correlation between the patient's cells and the patient clinical status. What is the mechanism? So this is the graph you already know, the reduction of arrhythmias. This is the effect of SOICR. So anything that reduces SOICR has an effect of stabilizing the ranitidine receptor. And you can see that the known stabilizing ranitidine receptor drug indeed does so. So does carvedilol. Flecainide is an interesting question. There's a big, big debate in the literature whether it only works as an antiarrhythmic that blocks sodium channels or whether it stabilizes ranitidine. The Americans think that it stabilizes ranitidine. Silvia Perori and the Italians think that it doesn't. Our results support more the Italian theory. You can see that it reduces arrhythmia, but at regular dose has no effect on ALCR. At high dose, it has some effect on stabilizing the RA. But the majority of effect is probably through sodium blockade. Okay, going back into the regenerative medicine, the second hurdle is making different heart cell types. To repair an infarct, of course, you want ventricular cells. You don't want atrial cells. So together with Gordon Keller in Canada, we are now trying to derive methods in order to derive the different heart cells types of the heart. You know, there's the SA node, AV node, conduction system, fibroblast, epicardial cells, endocardial cells. And we believe that within the next few years, we'll be able to generate methods to make each of the heart cells by itself. So we'll be able to make patient-specific pericardial cells, patient-specific atrial cells, and so on. This is one example in which we can make only ventricular cells or only atrial cells. You can see that the atrial cells don't express a ventricular marker, while the ventricular cells express them. You can see also that the ventricular cells have a ventricular-like action potential, and the atrial-like have an triangular atrial shape typical of atrial. And we also measured a current, which is only present in atrial cells, IK-acetylcholine, and you can see that indeed the black tracing, you can see the current in atrial cells but not ventricular cells. So this is important because we want to regenerate the heart, we want ventricular cells. What can we do with atrial cells? For example, model atrial fibrillation. So you all know what atrial fibrillation is all about. The problem that atrial fibrillation does not manifest in a single cell, so we had to develop more of a tissue-like. So now we're growing a few millions of cells, trying to generate something like a small atria in a dish. And we can see that it starts to beat synchronously like sinus rhythm. We can now pace fast, and within this atrial tissue, we can even induce re-entry seekers. You can see these are called rotors. It's a big thing now in atrial fibrillation. People think that this is what drives atrial fibrillation. And now we can test what drugs do to it and how to ablate it and so on. We further do this by using an optical mapping system. So now we can study how electrical activity propagates in these atrial or ventricular tissues that we create. This is how a rotor looks like. So now we can test the effects of drugs and interventions and understand the biophysics of these rotors. We can also make pacemaker cells. This is a work for Stephanie Sprotze. So we can make SA node cells. What can SA node cells help us? For example, maybe use as an alternative to biological pacemakers, as alternative to electronic pacemakers. So we studied the difference between pacemaker and ventricular cells. When we put SA node cells on top of ventricular tissue, you can see now that the SA node cells start to pace the ventricular tissue. In contrast, when you put ventricular tissue on ventricular tissue, you can see that the beating rate is really slow, and these ventricular cells are only passively activated. So at least in vitro, they can pace the ventricle. What happens in vivo? So we transplanted either 2 million SA node cells or 2 million ventricular cells into the heart, into the rat heart. 2 weeks later, we took out the heart and studied it using a Langendorf preparation and optical mapping. We injected it into the apex of the left ventricle. So initially we were disappointed because if you look at conduction, it starts the red area, then yellow, green, and blue. It starts from the septum, not from where we transplanted the cells. But this is not surprising because the heart rate of the rat is about 300 beats per minute, much faster than the human SA node cells, so the rat dominates. So we had to slow down the rate. We did it by developing a pharmacological cocktail that induces transient AV block that lowers the heart rate. And you can see that now activation starts here from the apex where we transplanted our cells. You can see it here in this map. So this summarizes it. We have sinus rhythm coming for the rat. We induce AV block. There is a new paced rhythm. All of the beats originating from the SA node-like cells that we injected. What happens when you put ventricular cells? Rat conduction, you induce AV block. There is either nothing or a junctional escape rhythm coming from the rat conduction system. Okay, so this shows that maybe we can use this cell as an alternative to electronic pacemakers, and also very importantly that the ventricular cells are not arrhythmogenic. This is important when using them for repair of myocardial infarction. How are we moving to clinical trials? So we are now into large animal. This is a study published recently from Seattle, and one of the PIs, Michael Laflamme, has recently moved to Toronto, so he's part of our group. In this study, they injected 1 billion human heart cells into an infarct model of monkeys, and the results are very nice. The green cells are the human cells. Look how nice they integrate and repopulate and remuscularize the heart, and they can regenerate up to 70% of the infarct. And these cells also integrate with the existing tissues. So the fact that we can make many cells and the fact that they're encouraging results in large animals makes us to believe that we can go into humans in a few years. Now, people with heart failure don't have only mechanical dysfunction. They might have ventricular arrhythmias, as you all know. They might have bradyarrhythmias, and they might have a mechanical desynchronization. So in the last couple of minutes, I would like to show you a new technology called optogenetics. Tries to address this. Optogenetics is a technology that transforms neuroscience. It all started when scientists realized that algae move towards light or away from light due to the presence of light-sensitive proteins in their membranes. A typical protein is channel rhodopsin, which is a cationic channel that opens following exposure to light. So researchers now, using genetic engineering, express this protein in the brain of animals or in neurons in vitro, and now you can control nerve activity with light. You can either activate it or suppress it with a different set of proteins, so you can really control brain function. So we wanted to see whether we can take this technology into the heart. So to this end, we delivered the protein, the transgene encoding for the protein, to the apex of rat hearts. 2 weeks later, we took out the heart and we studied them in the language of preparation. You see that this heart is beating happily ever after. We now shine flashes of blue light. And you can see that every flash of blue light introduces a paced beat. So the blue light activates the light-sensitive protein. Sodium rushes in, generates an action potential, so this area now functions as a pacemaker. Again, every blue light results in a paced beat. So you can, similar to electronic pacer, vary the beating rate, the pace rate. We can also do it not outside the body, inside the body. Here, we're doing it inside the rat chest. Again, you can see the heart beating. We flash light, and we pace the heart with light. We then wanted to see whether we can do a more advanced pacing, for example, a CRT based on light. The problem with CRT that we are limited in the number of electrodes and their location, but light has no boundaries. We can shine wherever we want, so we can activate the light from any number of sites. Here, for example, we wanted to see what happens if we shine simultaneously from 3 or 4 sites. So we compared the electrical activation of the ventricle here in 3 sites. So initially, this is how sinus rhythm looks. It's very, very synchronized, as you all know. See, the entire ventricle is activated simultaneously. We then pace the right ventricle in order to simulate left ventricular desynchrony. You can see that it takes longer to activate the ventricle. And then we activate the ventricle from 3 sites simultaneously by light. Again, this is very synchronous. If we summarize the results, sinus, small number, sinus rhythm, very fast activation, RV pacing, desynchrony, optical to genetic pacing for multiple sites, resumption of activation time similar to sinus. And last, we wanted to see whether we can do the opposite. Can we stop electrical activity in a similar way that we do during electrical defibrillation, where we induce continuous depolarization simultaneously, but as you all know, this is painful. Light should not be painful, so we wanted to see whether we create a model, do a fibrillation with light. So we first started in a dish. To this end, we use a disease model of the short QT syndrome. This is the healthy, where you have normal rhythm. These are ventricular cells. Here in the short QT syndrome, you can see we can have these reentry circuits developing. So we then wanted to see whether we can defibrillate this so-called VF. Here you see it's very irregular, almost like VF in the dish. We give a light, it stops, and then we see a resumption of normal activity. Can we do it also in vivo? And this is the last slide. So we created an infarct. 2 weeks later, we took out the heart and induced ventricular fibrillation. And again, you can see that it's beating sinus rhythm. We do program with electrical stimulation, and it starts to fibrillate, as you can see now. It's fibrillating. And now it's in fibrillation. We will try to shine blue light like electrical shock. And as you can see in a second, it defibrillated it, and it goes back to sinus rhythm. And with that, I would like to end and thank the people in my lab that did all this work, our collaboration in Israel and the world, and I'd like to thank you for your attention.